the sea ice, the base of the food web is the algae, the ice algae, which grow generally at the bottom of the ice, so where the ice meets the water. And that's because the algae need two things mainly. They need sunlight, which comes in from above, and they need nutrients, which come from below. So that feeds the bacteria within the ice, and we see a lot of different types of bacteria that you find either rarely or not at all in the water underneath the ice. And it also feeds the water column and the benthos, the sediment organisms below. And so there's a very high abundance of organisms in those regions because of this connection between the ice and the sediment. We study psychrophiles, which are extremophilic organisms that live at cold temperature. And psychrophiles live only at cold temperature, so if you bring them to room temperature, they die.
Here we go.
What we do is we get ourselves onto the ice. And once we get to the ice floe, we pick a suitable spot, usually somewhere fairly level, and we have an ice auger that takes a core that it's about this big around and up to 1 meter long. We have several extensions so that once we pull out 1 meter, we can go down again. And the longest one we've got so far is 4 meters long. The ice that we're studying is only a couple of years old. So maybe the oldest ice that we've collected so far on this expedition is 5 to 10 years old. And that's most of the ice in the Arctic is less than 2 years, in fact. So then once we get the ice back to the lab, we take it into the freezer room so it doesn't melt while we're working with it. And we lay it out and look for any variations in it, whether there's air bubbles usually in the surface or whether there's a band of algae growing at the bottom. Then we start cutting it into sections. So we'll cut maybe a 5-centimeter section to melt for the ice salinity. With that, we can learn about the history of the ice. And we cut other sections to look at the biology in the core. So for those, we will add filtered seawater. So we'll take natural seawater and we'll filter all the organisms out of it and then use that to melt this ice so that when the organisms come out of the brine channels that they live in, they're not shocked by the low salinity. So then once we've melted all the ice, which usually takes overnight, then we put it through a very small filter that has a pore size of 0.2 micrometers. So a human hair is about 100 micrometers across, so this filter catches things that are 500 times smaller than a human hair. We catch all the bacteria on these filters, and then we freeze them at ultra low temperature to preserve them until we get back to shore. And then once we're on shore, we'll take them and extract the DNA from them and then sequence that on a DNA sequencer. And from that, we can learn about the diversity and the evolution of these organisms. We know that the ice is decreasing, but we don't know what effect that is going to have on the biology. There's different options that people have considered. One is that the algae in the surface water will be more productive because there's more light, because the ice isn't blocking the light. And you can imagine that would rain down and perhaps increase the total productivity of the system. Or they might decrease in productivity because there will be more meltwater on the surface, stronger stratification, which means that there's less nutrients coming up from below to feed the algae. Then we would actually have less productivity, and that would cause a decline in the total ecosystem. And both of those are options, and we don't know which one is correct. So what we're doing is finding out what the system is like now so that we can track it as it changes into the future.